Assalamu alaikum, dear students. Today we will talk about the cell and its function. We will talk about the types of cell as well as the cell theories and those scientists, those biologists who have contribution in the cell theory. We will discuss both the modern cell theory and the initially proposed cell theory. As well as we will also discuss some basic and common function of the cell at the end of this lecture. So what are the objectives of today's lecture? In this lecture, we will talk about define cell and its types, state the cell theory, describe the organization of cell, explain the physical structure of cell, enumerate protoplasm and its component, and draw a neat diagram of cell and its organelles. So according to a great scholar, the great acts are made of small deeds. Similarly, the great and the huge animals and living species, they all have made from a very small structure known as the cell. The basic living unit of the body is the cell. Each type of cell is specially adapted to perform one or a few particular functions. The entire body then contains about 100 trillion cells. अब यहाँ पे अगर हम स्पेशली अडॉप्टेड वर्ड को देखें, इसका क्या मतलब हुआ? हर एक सेल की स्ट्रक्चर जो है उसकी फंक्शन के मुताबिक जो है अडॉप्ट हो गई है। अब स्केलेटल मसल को देखिए तो स्केलेटल मसल जो है ये कंट्रैक्ट होती हैं, शॉर्ट होती हैं और फिर रिलैक्सिंग के दौरान ये लेंथन हो जाती हैं। अब यहाँ पे जो स्ट्रक्चर इसको कंट्रैक्ट यानी शॉर्ट एंड रिलैक्स कर देती हैं वो sarcomere है जो जिसके अंदर thick filament and thin filament होते हैं, the myosin and the actin, और साथ साथ दूसरी भी कुछ इस तरह की फिलामेंट्स होते हैं। सो ये तमाम स्ट्रक्चर जो हैं किसी और सेल में नहीं होती, ये किसी एक सिर्फ और सिर्फ स्केलेटल की एक मायोसाइट में होती हैं, क्योंकि ये उसकी डिमांड होती है बिकॉज ऑफ दिस एक्टिन एंड मायोसिन there is contraction of this muscle, there is shortening of this muscle. Similarly, अगर हम एक न्यूरॉन को देखें which is a nerve cell, इस न्यूरॉन की जो पार्ट्स होते हैं which is the dendrites, the cell body की जिसको हम सोमा कहते हैं, उसके बाद उसके अंदर एक न्यूक्लियस होता है और उसके बाद एक लेंथी ट्यूब निकलती है which is known as the axon, जिसके अंदर फिर एक्सोप्लाज्म होता है that protected by the exolemma, और ये जो है कंडक्ट करता है एक जगह से दूसरी जगह तक इम्पल्स को इन इलेक्ट्रिक इम्पल्स, यानी ये मैसेज जो है ये ब्रेन की तरफ लेके आते हैं और एज वेल एज ब्रेन से बाहर पार्ट्स को भी मैसेज लेके जाते हैं। तो स्ट्रक्चर ऑफ दैट न्यूरॉन इज क्वाइट डिफरेंट फ्रॉम द स्ट्रक्चर ऑफ द स्केलेटल मसल। इसी तरह अगर हम स्मूथ मसल सेल का या कार्डिक मसल सेल का किसी भी सेल उठा ले और इसको देख ले तो उसमें जिसके अंदर उसकी स्ट्रक्चर होती है ये डिफरेंट होती है बिकॉज all these type they have specially adapted to execute a very specific function. So what is the cell? A cell is a basic living structural and functional unit of the body, and the scientific study of the cell is known as the cytology. And what is the cell physiology? That is actually the scientific study of the function of the cell is known as the cell physiology. So if we are going to study the structure of the cell, this is known as cytology. And if we are studying the function of the cell, then cell physiology. We are here to discuss the cell physiology. So we will not give so much concentration and focus on the structure of the cell, rather than we will give more and more concentration on the cell function. The cell can be divided into three peripheral parts. the principal parts, and these are the plasma, which is also known as the plasma membrane, also known as the cell membrane, cytoplasm, and cytosol. They both are present inside the plasma membrane. The second principal part is the organelles. Different organelles like mitochondria, Golgi bodies, ribosome, and different organelles, but except the nucleus, we will consider and will study the nucleus separately. So these three principal parts, they are the plasma membrane, the organelles, and the nucleus. The history of the cell. Initially, the cell was seen for the first time by the Robert Hooke in the mid 1600th century, and he observed the cellular of Kirk, and in that he saw some row, row of empty boxes, and that's why he termed this chamber in this row of empty boxes as cell, because the cell literally mean a chamber, and these structure that look like the chamber, so that's why उनको सेल नाम जो है रॉबर्ट हुक ने दे दिया. उसके बाद 1838 में मिथियास श्लाइडन आ गया और उसने एक थ्योरी पेश की, उसने कुछ स्टेटमेंट पेश की. ही वाज अ जर्मन बोटानिस्ट बेसिकली, एंड उस शवांग के साथ उसने जो थ्योरी पेश की थी, उस थ्योरी की इनिशियल जो दो स्टेटमेंट हैं, ये श्लेडन और शवांग के हैं, जबकि एक तीसरा बंदा भी था जिसकी कंट्रीब्यूशन थी, वो हम आगे डिस्कस करेंगे. So he viewed the plant parts under a microscope and discovered that the basic unit composing different parts, plant parts, are cell, and that all plant tissue are composed of cells. The co-founder of the cell theory was also another person, the Theodor Schwann. He presented and proposed the cell theory for the animal in the 1839, just 1 year after the plant cell theory. So Theodor Schwann was a German biologist who viewed animal parts under a microscope and discovered that cell were the basic unit they are composed animal. This was a very important contribution since this extended the Schleiden cell theory in plants to animal, stating that all living things they are composed of cells. So the cell theory, the people recorded as on the slide have contributed to the cell theory, which state that all living organisms are composed of cell. They may be unicellular or multicellular. And the second statement of this theory was the cell is the basic unit of life. The cell arises from the pre-existing cell. This third component, the cells arise from the pre-existing cell. This statement was actually not by these two German biologists. There was another researcher. He was a biologist, but later on he became more interested, and he have more contribution in the pathology. He is also known as the father of the modern pathology, the Rudolf Virchow. अब Rudolf Virchow ने ये डिस्कस कर दिया, ये ऐड कर दिया कि सेल जो है ये पहले से जो मौजूद सेल्स होते हैं वही से न्यू सेल बनेंगी. So उनका एक लेटेंट फ्रेज भी कुछ इस तरह था कि एमिनो सेलुला ए सेलुला, विच मीन द सेल जो है ये न्यू सेल जो है ये प्री एग्जिस्टिंग सेल से बनेंगी. So actually three statement and three researchers, three scientists, they have contributed to the cell theory. All living organisms are composed of cell, चाहे unicellular या multicellular, ये एक बात हो गई. The cell is the basic unit of life, ये second, and the third one is the cell arises from the pre-existing cells. The modern cell theory indicates that energy flow occur within the cell. Hereditary information, which is the DNA, is passed on from one cell to another, and that is the reason once that daughter of a cell, they are similar to the parent because they're sharing the same DNA. All cells have the same basic chemical composition. So there are two types of cell, the eukaryote and the prokaryote. As we can see here in the eukaryote, there is a true nucleus. On the other hand, in the prokaryote, there is no such membrane bounded nucleus, and there is a scattered material known as the nucleide. Otherwise, the ribosome are present in both cells. Cell membrane is present in both cells. There is cell wall as well in the prokaryote, while there is no such cell wall in the plant eukaryote cell. And a flagellum is also present there along with the capsule. These both are also absent in the eukaryote cell. So these are the primitive and first cell type on Earth, and these are actually the bacteria are all prokaryotic cells. No membrane bound nucleus, and instead of that, uh, the DNA concentration is present in an area known as the nucleide. An organelle is not bound by membranes, only the ribosome. Ribosome is one of the organelle which is non-membrane bounded, so that's why ribosome is also present in the prokaryotic cells. On the other hand, the eukaryotic cells, they are advanced cell type. Nucleus is bounded by membrane, and they produce many membrane bounded organelles like the mitochondria, like Golgi apparatus, like endoplasmic reticulum, and DNA is present inside the nucleolus. This is the structure of the eukaryotic cell. You can see here, this is the nucleus, this is the nucleolus where the DNA is present. This is the rough endoplasmic reticulum. The endoplasmic reticulum, which have ribosome on its surface, is known as the rough endoplasmic reticulum. Here, this is the smooth endoplasmic reticulum. There is no ribosome on its surface. These are the smooth endoplasmic reticulum. There is no ribosome here. These are the lysosome. This is the mitochondria where the energy reaction occur, the powerhouse of the cell. These are the Golgi apparatus. These are the ribosome which are not attached to the surface of the endoplasmic reticulum. Microfilament. This is the plasma membrane, also known as the cell membrane. And these are also the microtubule, the centriole. So all these structures we will discuss each of them in the upcoming lecture. The cell is what is the composition of cell? So the term protoplasm, they include both the substance within and the cell membrane, and is the living substance of the cell. It can be differentiated into cytoplasm and the nucleus. What are the physical properties of protoplasm? So this protoplasm is a transparent and jelly-like material. The consistency varying from the more liquid, slightly gelatinous white of a fresh egg to that of the semi-solidified gelatin of jelly. Sometimes, if the viscosity of the protoplasm is more, it means that is more viscous, that will be called as the gel. But if it is a liquid, more liquid, and its viscosity is less, there is less resistance to its flow, then it will be called as a sol. Now, what are the chemical properties of the protoplasm? The chemical properties of protoplasm can be divided into inorganic and organic substances. The inorganic substances, they are water, which make up to 90% of the protoplasm. The other inorganic substances, they are the mineral salt, such as initial salt, and gases like oxygen and carbon dioxide. On the other hand, organic substances, they include the proteins, carbohydrates, lipids, nucleic acids, and enzymes. Enzymes are also proteins. Now, what are the function of the protoplasm? We will discuss various function of the protoplasm. The first one is the reproduction. The cells divide to form identical daughter cells. Function of the nucleus of the protoplasm. Reproduction, that simply means the division of one cell into two and two into four and so on. There is one term which is known as the hypertrophy. Hypertrophy is actually not increase in the number of cells, that is just increase in the size of the cell. While when the number of the cell is increasing to that from 2 it make 4 and from 4 it make 8, it is known as the hyperplasia. When the size of an area is suppose the skeletal muscle, if it increase in size due to the increase in the number of their cells, because their reproduction in their cell, so that will be called as the hyperplasia. Irritability, this is the very important for the cell, that when the protoplasm that being disturbed, being irritated by some foreign body or some stimulus, so it will give response. For example, the retinal cells in the eye, they respond to light. When the eyes are exposed to sharp light, there is constriction of the pupil, and when there is dim light or in the darkness, there is dilation of the pupil. So this such reflexes in the retinal cells in the eye to the light. Chemical, all these functions are carried out inside the cell. For example, the respiration inside the mitochondria. Excretion, cell must get rid of excretory waste. Suppose, for example, in the previous slide, we discussed the chemical reaction respiration. So there is a waste product of the respiration, the carbon dioxide. Now the cell have to get rid of this carbon dioxide. So they usually diffuse out of the cell through the cell membrane, and the same it does against the carbon dioxide. The movement is exhibited by certain cells, not all cells. Um, for example, the unicells, the protoplasm of these cells has a contractile ability. Growth follows on cell division. There is an assimilation of protoplasm and increase in size. Physical structure of the cell. This is actually the structure of a cell in which there are different membrane bounded and non-membrane bounded structures. The outermost coat of the cell is known as the cell membrane. As we discussed earlier, it is also known as the plasma membrane. The cytoplasm, the blue color which is seen here inside that light blue line, this is all the cytoplasm. The cell organelles, these the endoplasmic reticulum, the ribosome, the smooth endoplasmic reticulum, and the Golgi complex or the Golgi apparatus, Golgi bodies, the mitochondria. These all are the cell organelles. The nucleolus that is in the middle, this is the nucleolus. Now, if we discuss the structure of the cell membrane, so it is a lipid bilayer in which it is made of the head and tail. The head is that is water loving, and the tail is water hating. It means water can easily, if there is need um, for the transport of water through this cell membrane, so it can pass through, because the head that is water loving and it is called as the hydrophilic, while the tail that is the hydrophobic. These two are the membrane proteins, also known as these are present from one end to the other end, so they are known as the transmembrane. While these membrane protein which invigilate inside the plasma membrane, but they never reach till this end. So these are known as the monotropic protein. These are known as the bitropic protein, and other protein which have many fold and many, they span multiple times across the membrane. Three or more than three or many, specifically they used for the transport, so they are known as the polytropic. This membrane, this is actually you most of the cases they are the receptors, and while the biotropic and specifically the polytropic, they are the transporters, the carriers, and the enzymes as well. As we will discuss in the upcoming slide, this is actually the cardboard red part which is connected with the protein. So this is known as the glycoprotein. Similarly, the cardboard red they can also be connected with the lipid, and this is known as the glycolipid. These protein which are present at the surface of the this lipid bilayer, these are known as the peripheral protein, also known as the extrinsic or external protein. These two are example of the this is monotropic and the bitropic, and both are example of the integral protein, also known as the intrinsic or internal protein. So this is actually the lipid bilayer. We can have the head and tail in these structures. These two are the peripheral protein. This is the integral protein. This is these all of these three. These are the bitropic protein. This is again the peripheral protein. The glycolipid, the glycoprotein. This is the oligosaccharide, which is a carbohydrate, and this is the integral protein because it is invigilated inside the lipid bilayer. These two are the leaflets of the lipid bilayer. This is the phospholipid bilayer. So this head and this is the hydrophilic, which is actually we can call as the water loving or hydrophilic polar head, while the middle portion, which is actually the tail, this is the hydrophobic core, or also known as the water hating center or the water hating core. The structure of the cell membrane composed of phospholipid bilayer with a college of many different proteins, lipids, and carbohydrates, as we discussed in the previous slide. A phospholipid is composed of one glycerol molecule, two fatty acid, and one phosphate group. This structure causes the hydrophilic and hydrophobic region. You can see here the structure of the phospholipid. This is the phosphate group. This is the glycerol, and these both phosphate and glycerol they make the head of the phospholipid, and that head is actually the hydrophilic, while the tail is made up of fatty acid, both the saturated and unsaturated. The unsaturated mean which they are having the double bond, and saturated mean they don't have any double bond. So the tail is the hydrophobic and the head is the hydrophilic. So we can see here as if there is contact of water with the tail, so that will repel it. There is their water header, while on the other hand, the head of that phospholipid, this is water loving. This is the hydrophilic head, while the tail is the hydrophobic, and it is known as also known as the water heading. You can see here the membrane protein. There are two types: integral protein that insert into the membrane, also known as the transmembrane protein. Peripheral, they are protein attached to the surface of the cell membrane, and here we can see here the other three types. These three types, the monotropic, the bitropic, and the polytropic, they are the example of the integral protein. If a protein is partially integral, it means partially inserted inside the membrane, it will be called as the monotropic. If the integral protein they completely span from one side to another, it will be known as the bitopic. And if there is a integral protein which are have multiple time at span across the cell membrane, it will be called as the polytopic. A main function of this membrane protein is not only the transportation, but the most important one is the transportation. It can be enzyme, a receptor site. Usually the monotopic, they are the receptor. Biotopic, they are mostly enzyme, and Polytopic, they can be enzyme and the transport carrier, both can be. And cell adhesion and attachment to the cytoskeleton. So these were the function of the membrane protein. The carbohydrate, usually branched molecule at 15 or less sugar unit. Some of the bonded to lipid are also known as the glycolipid, and some are bonded to the protein known as the glycoprotein. The glycoportion produced outward, giving a loose carbohydrate called the glycocalyx. Due to the negative charge, it repels either negative charge particle. This is actually the property and the function of the glycocalyx. There is through the glycocalyx there is cell-to-cell recognition, and through the glycocalyx, that glycocalyx that act as a receptor subtransfer hormones such as the insulin, and through the glycocalyx there is also prevention of the entry of the pathogens inside the cell. So this is the structure of the lipid bilayer, the cell membrane. We can see here this is the extracellular fluid. This is inside of the cytoplasm. This is the cytoskeleton which is present inside the cytoplasm. This is the peripheral protein. This is the integral protein. These are the cholesterol, the lipid. This is the lipid bilayer. This is the glycolipid. This is the glycoprotein. This is actually the carbohydrate portion. And that was end of today's lecture. Thank you so much to all of you. Have a nice time.